So replication theories. To help support your understanding of this concept, I'm going to give you a link to a Khan Academy study page. They do a really good job. It's actually overkill. They have a lot of information. I'm going to explain the replication theories in this quick little lesson here. And but there is no PowerPoint that I've created that has this particular technique outlined for you. So use the Khan Academy video and a couple other videos I'll link to a Google Doc for you to use. So we know that DNA is able to replicate itself. The DNA is able to replicate itself almost perfectly. We know from grade 11, if you have one mistake, one chromosomal mistake, you can end up with Downs. In grade 12, we're going to learn that one nucleotide out of place can give you sickle cell anemia or cystic fibrosis or many other diseases. So it's very important that the DNA is replicated accurately. And if you were to type 60 words a minute and type out the entire human genome, I think it takes something like 50 years or something crazy like that if you type from 8 to 5. So there's a lot of information. So this DNA has to be replicated accurately and quickly. So scientists were trying to figure out, before the microscopes were effective and before a lot of our very sophisticated newer biochemical techniques, how DNA replicated. So there's 3 theories. Here's our original DNA. Right? And our original DNA, it can replicate one of 3 ways, potentially. And so one of these is correct. Here's original DNA here at the top. Let me just get a pen tool. I'm going to use my board here. So if I'm looking down, I'm looking at my board. Right. So this is my original DNA. And so this original DNA is a double helix, 2 strands, anti-parallel. One of the potential replication theories is that it could be dispersive, where basically the entire strand here explodes, separates, and the new DNA is incorporated, and you end up with 2 brand new strands. That's known as dispersive. In the other model, known as semi-conservative, let me just grab these words here so you can follow along. So dispersive, right? And semi-conservative, it's suggested that each strand here separates and it becomes a template for the new strand. Right? So that's a possibility. That's semi-conservative. And in the third potential model, it's called conservative, where the original DNA stays intact and it produces a copy of itself. And so there's one original and one new copy, and that's known as conservative. So one of these 3 is correct. It's either conservative replication, semi-conservative, or dispersive. And so this scientist pair, the scientist duo, Messelson and Stahl, and if you look them up and Google it, you'll find hundreds of pages with lots of animations and lots of videos explaining this. And I've picked a couple of good ones that are written at a good high school language to help you with the concept. But I'll try to explain it using this whiteboard here. So Messelson and Stahl, they had this ingenious experiment. Nitrogen, like many other elements, comes in a variety of different forms. These isotopes can be heavy or light depending on the chemical structure. So we have N15 and N14. N14 is our typical nitrogen. So here's our original DNA. Okay? And this original DNA is made up of N14. So remember, the nitrogen is incorporated into the nucleic acid structure and has a certain mass. I can place this DNA into a test tube. So I place it into a test tube, and I'll use my pen here. Hopefully it'll work. If I have all those lines on the page in case my pen doesn't work and I need to use a different technique. So let's go with a line here. I'll go with a pen tool, actually. I'll use blue. And so inside this test tube here, it gets spun in a centrifuge. And based on the density of the chemical, the compound, it will either go to the bottom of the test tube or it will go to the middle or stay near the top. So this whole thing's full of kind of a DNA liquid. So it's spun around, and it's found that all the DNA is sitting right here. So now this DNA is allowed to undergo a round of replication. So one round of replication. But instead of N14, which is what we have in our original DNA, the new DNA is going to have access to N15. So this is a heavier isotope of nitrogen. So any new DNA that's created will have N15, and only the original DNA will have N14. So what they did is they added a whole bunch of N15, they had a DNA polymerase, we'll talk about that a bit later, they let it replicate, and then they spun it around in the test tube again. And when they spun it around the test tube again, what they found was that there was one band right about, let's make it black. There was one band right about here. So that gives us some information. So remember, we have 3 different models. I'm trying to figure out which model is accurate. Is it the dispersive, the semi-conservative, or the conservative? In the dispersive, after one round, if it were successful, you would have this DNA that's made up of N14 and N15 equally. Right? So this would produce one band. So that means this is still a possibility. If it was semi-conservative, you'd end up with one whole strand that's light, original, and the N14, one whole strand that's heavy, the N15. And so this DNA double helix would be also one strand that's kind of intermediate, like N14 and a half. So both this and this would produce one band. This one, however, it produces a whole DNA that is 15, N15, and a whole DNA that's 14. So this one here, conservative, it would have produced 2 bands. But when we look at our results here after one generation, there's only one band. So what that allows us to do is allows us to rule out conservative replication. This is not a possibility, gone. But we don't know if it is semi-conservative or dispersive. All right, so what do you do next? You run it through several more rounds of replication. And what they found when they ran it through another round of replication is that there was a band here and there was a band here. And each time they ran it, that band that was higher up it remained about the same. And the band that was lower down, it got thicker, but it was always lower. So what does this tell us? So let's think about what the results are going to look like. So when we go to our possibilities, let's say that we have our original is N14. And then it is, dispersive. We'll go to the dispersive model first. So in the dispersive model, the suggestion is that this original DNA blew up into little pieces, and then the new DNA was incorporated, like we see here. So this blew up in little pieces, and we ended up with two brand new helixes, both made up of pieces of the old N14 and the new N15. If this went through another round of replication, then this original and this original and this original and this original, all these strands here would all blow up some more, and they would produce more dispersive. But each time, we don't have any more originals. That blue would be getting less and less and less as it gets dispersed across more of these. So this one, the single one here, if I was to focus on just this here, and I replicated it, it would produce 2 more dispersive. So if dispersive were true, then I would actually end up with 1 band. That band would be heavier, but it would still be 1 band. Okay? That does not hold true based on this model. Based on this model, we actually have 2 distinct bands. 1 band after 1 round, and then 2 bands after 2 rounds, and then 2 bands after many rounds, with the bottom band being much thicker. So this doesn't fit that model. So let's move that out of the way here. So let's go to semi-conservative. In semi-conservative, here's your original DNA. The idea is that each of these would separate, and the new DNA would be incorporated. And so this would be like N14.5, and this would be N14.5, which is what we saw, just like the dispersive model. It's a possibility. So then what would happen next in the next round? Well, in the next round, I'll just focus on this one here. In the next round, each of these would become parents. So this would separate, and this would separate from each other. And now you get the incorporation of only new DNA. And now what do you have? Well, I still have this one here, which is kind of the medium N14.5, N14.5. And then I got this new group here that's only N15, right? So this semi-conservative, after 2 rounds, would produce 2 bands, a medium band and a heavy band. And if I kept on doing this over and over and over again, I'd always have the same medium bands here, because I only ever have these 2 original DNA strands of light nitrogen. And then these new bands are always forming. They would always be more heavy nitrogen. And so I'd end up with a thicker band here each time, getting thicker and thicker and thicker. So Messelson and Stahl were able to use this technique to determine that the semi-conservative replication, this one right here, that is the most accurate and the correct way that DNA replicates. And it makes sense. A template, a perfect template that the DNA is then able to copy perfectly. And if there is a mistake, then it can fix it. That is the replication theory determined by Messelson and Stahl. So now you can add Messelson and Stahl to the group of scientists that I asked you to know for this unit. So you have Mendel up to Watson and Crick, and somewhere in there you're going to place Messelson and Stahl. And they're the ones who determine that DNA replicates semi-conservatively.